So this session is really focused on advances sort of beyond the models that we've been talking about. What Jacob's going to talk about, he's a data scientist, and he's going to focus on the use of machine learning and synthetic data, things we touched on, but really are catalyst accelerators for what we all do here. And I'd like to invite him to come up and share his perspectives on how this is going to affect the field. Jacob.
Thanks, Steve. Thank you. All right, thank you very much. And it's a pleasure to be here. Steve is someone who I think gave a talk at one of the first retreats when I joined Medidata, and it was really incredibly inspiring. And then he has tracked me down over the years in coffee shops and actually caught me in a coffee shop in Berkeley and invited me to come, so that's why I'm here today. But I'm really excited, and I think one of the themes of my talk is going to be talking about collaboration, and I think for me, one of the most exciting things about forums like this is seeing how many people are working on these problems and working collaboratively to bring better therapies to market. So in the interest of sort of playing to the engineers, I want to put together a bit of a system diagram that will inform the talk. So just a general idea of a data-driven model of healthcare, and this went back to when I was in med school. They basically said you have clinical evidence, things that are in clinical trials, you have your training, and then you basically have the patient in front of you, and that is going to inform what treatments you give. And I've also added the concept of context, because obviously if that's an emergency department or a hospital or a clinic, exactly how you're going to treat that patient might be different. And I'm going to break this up a little bit differently and say, and this will sort of begin informing the approach from like a machine learning perspective, which is really what you have is knowledge. And I'm going to say that that's a combination of the clinical evidence or the data back there, plus these models. Medical training is a version of a model. I'll be a little bit more specific about that. And then you need to apply that data in a specific context, and that's given information that is specific to where you're going to apply that. In the world of sort of LLMs, this is the first bit is the training stage, the second bit is the inference stage, and then you have this concept of feedback, which is the treatment updates the patient's state and so on and so forth, and knowledge actually moves a bit more slowly than the patient context. So if we focus on this concept of medical training, I think for this audience, it really is better to think of this as models. These are systems. These are understanding of how physiological systems interact, of how molecules interact with cells, how those cells scale up to the body, and that's sort of the stand-in for that part of the block diagram. And then when we think about the context, this really has to do with care delivery. So from a data perspective, a lot of that information is going to live inside of electronic health record systems, inside wearable devices, inside of other sort of diagnostic devices that are collecting information about the patient. And what I'm really going to focus on, because this is where metadata spends most of its time, is this part of the puzzle, which is really kind of like the ground truth that we want to use to inform the models that ultimately drive that decision making. Okay, so why do we focus there? So metadata has been around for about 25 years. The biggest, most successful product we have is an electronic data capture system that's collected data from, the slide keeps on being updated, but 36,000 trials and 11 million patients. So it's an immense repository of information that's been collected in clinical trials, and because that data is collected in a very specific hypothesis-driven way with very strict quality controls, it's an extremely valuable source of information about which medical treatments are going to work for which patients. So I'm going to talk about how we sort of take something like all of that data and think about a particular therapeutic context, and then use some of the tools that we've developed over time in order to solve problems that are both extremely difficult to solve and very important for patients. So about 5 years ago, we started focusing on CAR T cell therapy for cancer. This is a very expensive personalized medicine for cancer. The reason we focused here is because we realized that most of the companies that were developing these cell therapies had very little information from humans on which to develop those drugs. And so at the time, this slide I think is a bit dated, but it's from 2022. Most of the companies that were out there that were in the clinical stage had data on less than 15 patients. There were 3 companies that had commercial products that had a few thousand patients worth of data. That's the number of patients who'd been treated at the time. And Medidata had about 3,500 patients worth of data sitting in our systems in aggregate. And one of the real beauties of metadata is that for about 15 years, we've been very proactive about engaging our sponsors in data sharing collaborations where they actually allow us to pool and then utilize some of that data for secondary use under specific conditions. So the prerequisite to being able to build a virtual twin of a CAR-T patient, a patient who would receive this sort of therapy, is access to all of this data. And this is really a collective action problem. So when we engage in data sharing with pharma, that's under 3 different sort of core values, I would say. One is the concept of generosity, which is that data in clinical trials is, for the most part, given to the sponsors who collect that data by patients. Much of the intellectual property that goes to develop these drugs is developed in the public domain. So there is a real sense of this ecosystem and sort of knowledge flowing freely throughout it. The second is that data can be shared insofar as it is stewarded, it's sort of taken care of. We don't want to be thoughtless or careless in how we share that data. We have equities to protect in terms of intellectual property, in terms of privacy. And then we have to make that data useful or valuable, because people won't seek to share their data unless they think that there's value either for them or collectively for the broader community. And what I'm going to really focus on from a Gen AI perspective is these last 2 pieces, which are about stewardship and enablement. And the first one will be about stewardship, which is really, in the context of building up these virtual twins, we needed a way to produce data sets that were going to be safe and private, so that the patients who contributed to that and the sponsors felt comfortable sharing all that information. So the real challenge is that the sponsors that needed this data didn't have it, and the sponsors that had that data, it was locked up in their silos. So the question is, how can Gen AI solve that problem? So I'm going to go through a bit of a thought experiment, but this sort of speaks to one of the key pieces of technology we developed in metadata, which we call Simulants. So if you had a large corpus of images or something like that, several billion images, there are pretty well-established methods for producing synthetic images based off that initial source data distribution. But that's really based off the fact that you have many more images than you have degrees of freedom in the data set. You have maybe 10,000 pixels, and you have a few billion samples. And that is something that's very easy to train a machine learning model on. The problem with clinical trial data is that it is extremely small, and it is fairly sparse. So every single patient, there's maybe 100 patients in a trial, there's maybe a few thousand different pieces of data collected on that patient, and roughly the degrees of freedom in that data set are equal to the number of samples. And the problem is that when you try to approximate a sparse distribution of data, you have a situation where outliers that may represent true variation in the data set and the noise in the underlying data set are roughly the same magnitude. So you may have patients who are outliers because they're likely to have a certain adverse event. Those are outliers, but they're clinically meaningful. You also have noise in the data set. They're of a similar magnitude. And the problem is that the noise is something you want to eliminate from your model, but the outliers, that sort of signal that's coming from the periphery, is not something that you want to lose because you're approximating the distribution too coarsely. And in a real simple way, this is the problem with training a very generic architecture like a transformer on this type of data. So one possibility is you would upsample the data sets. You just create many copies of the individual patients. Now this has the benefit that it reduces the noise, but you end up with this very jagged distribution because basically you are suppressing the noise and suppressing the outliers in exactly the same way. We developed a method, a data augmentation approach that is, it's a bootstrapping approach where we resample the data set based off of finding local neighborhoods of patients who are very similar to one another and recombining their features in order to produce new density in between the true samples. And what you get when you sample the data this way is a smoother distribution with lower noise, but you're still able to capture a lot of that meaningful variation that represents the patient population. And for those of you who are kind of aware of AlexNet and some of these early machine learning systems that were trained on images, those training data sets, they actually took the images and they rotated them in different ways and they flipped them and they sort of went through all of these sort of geometric perturbations of those images, because the idea was a face, whether it's flipped in a mirror image or it's rotated, is still a face. So from the perspective of that machine learning task, they could produce 4 or 5 samples out of a single data point. And this is a very analogous procedure, basically. We are taking clinical trial patients and we are producing very plausible twins of those patients within the local neighborhood. Very similar to what they did to train AlexNet. And so the underlying algorithm is basically this. We fit the data to a very low-dimensional manifold. We identify nearest neighbors. We recombine their features. We do some interesting things to take advantage of the correlation structure of the data. We've also been able to generalize this so that whether the data is represented as a vector, as a sequence, we can do a very similar method. But this is Simulants. Now, what's the benefit of this? So the benefit, this is the paper that we published in 2022 in EMEA, but the benefit is that the black line here is the source data, the red line is the synthetic data. So from the perspective of some of the canonical metrics of similarity, so up top we have correlations, we have a PCA projection, we have a bag of words overlap sort of histogram, as well as from some clinical trial specific analyses. So here we have an overall survival curve and a progression-free survival curve. The real and the synthetic data are very, very similar. But the benefit, and I won't go too much into exactly what these metrics are, but the blue line here, the blue line across the top, is a measure, the y-intercept is a measure of how private or how much privacy loss there is based off just deleting the personally identifiable information in that data set. So basically, if you just delete the unique patient identifiers, it turns out that you are still revealing a fair amount of private information, roughly 85% of the private information, when you've simply anonymized the data set. But what you see with the synthetic data, the orange line and the green line, is we're able to, the y-intercept is very close to 0. So basically, despite the fact that the data contains almost exactly the same information, there's almost no privacy loss in sharing that data set, which means that if I gave you that data set, and I gave you 2 patients, and one patient was in the data set and one patient wasn't in the data set, you would be almost incapable of determining which of those patients was included in the source, in the original training data set, roughly. So the idea is that we've been using Gen AI here in order to produce a data set that has very similar information, but almost no of the private information that we want to hide. So this is a really sort of interesting feature of these sorts of algorithms. So what's the benefit? So now we've sort of solved our collective action problem because we have all these sponsors. They're able to provide data. They are each protected with respect to privacy. And through the magic of mathematics, we end up with a data set that is a clinical twin of the real data set that is both safe and also contains essentially exactly the same information. And in the world of my block diagram, this is like the raw materials for your virtual twin, right? We've basically produced a bunch of bricks and wood, right, that you can build your virtual twins out of, and those are very safe. We've removed all of the sort of contaminants from those building materials. So now we talk about enablement, because the next thing is, and I actually had an analyst ask me sort of like, what is the sort of cost associated with producing a virtual twin? And my answer was that the cost has to do with how many questions you feel like asking. Because to a certain extent, we're able to produce very good facsimiles of the underlying systems, but then the question is you have to interrogate it many different times. So the question is, how could we make the interrogation of that data more scalable? So we went through and we started working on patient journeys. So this is kind of a patient journey that runs from diagnosis. So this is a clinical trial patient. So it typically would begin with like a relapse of the cancer, enrolling in the clinical trial. They undergo apheresis, manufacturing of the actual cells. They're manufactured from the patient's own cells, treatment. There's a management process, like side effects that happen over the course of the trial. Then quantifying the response, and then hopefully reaching some sort of cure and the termination of the trial. And over the course of this journey, we began working on individual use cases that looked at either patient characteristics at baseline that were predictive of their outcomes, that looked at ways that you could manage the patient when they had certain types of immune responses that could end them back in the hospital and compromise the course of treatment. And the benefit is that, like, rather than sponsors working with 15 patients or 100 patients, like now we have basically a machine that can produce as many of these patients as we want. And it actually has a tremendous flexibility in terms of being able to look at patient subpopulations, variants within that population, perturb it in ways that you wouldn't even be able to do with real patients. Now, we've taken this work and we've worked with a whole bunch of different customers, and that's one of the ways that we sort of scale our impact, right? We go out to the pharma companies. Now, these are all companies that participate in our data sharing and actually contract with us for the AI and the AI services. So we work with them. We work with the CROs. We work with the smaller biotechs as a way to make these sort of insights more available and accelerate the development of these sorts of therapies. One of the other things that we do, which is, and Sheila's here, she leads the Research Alliance, and one of the most fun parts of what we do is we work with academics, because one of the things we learned is that, like, you have this great tool, which is this virtual twin of a cell therapy patient. The real thing you have to go is find all the people that have the questions to answer and actually the energy to go and answer those questions. And so we've gone and found a really amazing group of collaborators from across the industry, many clinician scientists, some machine learning researchers, and they have all been collaborating with us to interrogate this data and to find all sorts of new and interesting things. So in the context of the Research Alliance, we've been able to publish pretty extensively on CAR-T and then some other indications. It's one of the ways that I think it's these twins, these things are really only valuable if they're used. And I think that one of the most important ways in order to build things that will be commercially successful is actually to show that they have really high value in use. And working with the academic community has been an amazing way for us to do that. Now, as we've sort of expanded this work, we are now sort of creating characteristics of these patient populations that go beyond just sort of clinically how they behave. So we now understand how these patients behave from the perspective of trial enrollment and adherence. We call sort of more operational metrics. We also have information from sponsors about how they would like to configure their data collection systems, and we're able to feed these twins into those data collection systems so that when they're configuring the EDC or the ECOA system, they can actually look at data or test those systems with realistic samples drawn from these virtual twin populations. So here are some more examples, some of the most high impact examples that we've been able to do with this work. I'm actually not going to spend too much time on here because I want to make sure we get to the end. And then even more examples of how this evidence has driven faster enrollment, new endpoints, or situations where they've been significantly able to reduce the time and the cost of clinical trials by focusing on clinical populations that are more likely to benefit. Now, I wanted to make sure I got here before the end of the session because it's very relevant to what the previous group was talking about. So our future directions are really focusing on coupling our simulant system to more of the cutting-edge LLM systems, these transformer architectures, in order to produce more realistic patients at scale and also to produce more characteristics of those patients. Most of what I focus on so far is really structured data, and now we're thinking about ways to produce unstructured information, and obviously LLMs are extremely useful for that. And then I think we really shouldn't underestimate how much investing in the safety of these systems, also investing in ways that we can enable patients to consent for their data to be used to train these systems. We can help them understand the control that they have over their own information. We can also help them understand how they'll be protected by the systems, by the sort of protocols we put in place to steward the data, but also technically how the system obfuscates any information that they might want to keep private. The other thing I think that's extremely important is being able to work with regulators along with consortia like Steve has put together in order to advance the idea that this sort of information really is real, useful, actionable information that can be used to make well-informed decisions about drugs, and to make sure that ultimately the impact here is getting better treatments to patients sooner. So thank you all for the time.